My name is Labuda. I'm the CEO of Alzinova since October last year, actually. Come from a background in particularly pharma for the last 15 years, where I worked through R&D and commercial roles. As you know, Alzheimer is one of the big unsolved problems of this world when it comes to diseases, right? And we have currently more than 50 million people living with this disease. It's expected to increase actually over the next 25 years. We're going to have more than 100 million, and that's a conservative estimate, I would say. So there's a lot to do, a huge unmet need. We have the two first disease-modifying treatments on the market now, which is great, not only for the patient, but it's also proves that we are working on the right pathway, the amyloid beta pathway. There's still a lot to do. They are not as efficient as one would have hoped. So we have a huge opportunity here in the field. I mean, the amyloid beta pathway's involvement in Alzheimer has been there for quite a long time. And also we have known for the last 10 to 15 years that there's something else than the plaque that is actually driving the disease. We and many others believe that there's these toxic oligomers that actually are the most toxic things for the neurons, right? So a lot of people have known this and have worked on it. However, the treatments that we currently have, they are primarily focusing on removing the plaque, particularly Lilly's treatment, right? And they are quite successful in doing that. However, it does not translate particularly well to clinical efficacy when you look at cognition. So here we have a huge opportunity, and we believe that we have found the right peptide, you could say, that is actually driving this disease in its oligomeric form. Well, I think first, there's many things, right? If you start with the translation between in vitro models, in vivo models, and over to humans, there's a huge challenge in all of neurology, but maybe in particular in Alzheimer's disease. The animal models does not necessarily translate to what we want to see. So that's the first challenge, right? Now we're beyond that, you could say, because we have had our first patient studies. So good for us, and looking forward to the next parts of these trials that we are planning to do. So that's one of the challenges. The other thing is, of course, for us, it's been enormously challenging with the chemistry involved in stabilizing this peptide in a way where we can actually work with it. And I believe there's a lot of people that have tried this, but nobody has succeeded. So actually when Anders, my CSO, started with this back in 2011 in his PhD, they found that this might be a way forward, and it took a long time. And it's been a journey for sure for them to get to where we are now in patients for the first time. As I mentioned, I mean, it's been known for a while that this is what actually is driving the disease. And there's a lot of different proteins involved in this pathway, or peptides, you could say. There's fibrils, protofibrils, there's the monomers, and eventually they form this insoluble plaque that everybody knows are there in the brain, yeah? They have also identified a lot of different oligomers, and some of them have proven to be toxic in brain extracts, and others are not. So it's kind of difficult first to find the right one. And then, of course, working with something that is unstable and soluble is an enormous challenge, right? So to be able to do that has been a super challenge, but also thanks to working with people like yourself, I mean, it really helps us. So I guess this was in Anders's PhD where he started to see that this was toxic, right? And we were able to, in the brain extracts, able to remove these parts, and then we could see that the brain extracts were less toxic for the neurons. So, I mean, the evidence is there for sure, at least when it comes to the in vitro trials. And then going forward, we have now had our first phase 1b data, right? Where we actually see some very interesting data on the cognition. So that's exciting. So they have been focusing primarily on attacking, I mean, they are attacking the amyloid beta pathway, but maybe not the right part of it. So they claim that they are targeting everything, including the fibrils and the protofibrils and the oligomers. However, there's a challenge with this, of course, because the plaques are very abundant in the brain, right? So most of the antibodies that you passively inject will bind to the plaques. And then there will be very little left to actually bind to these oligomers that are sparse. There's not many of them and hard to detect. So with our treatment, we're extremely specific. We target nothing else, only these toxic oligomers. So that's the difference. I mean, so what we are doing, we are developing a therapeutic vaccine, right? So it's not a passive immunization like the others with antibodies. We are injecting the actual peptide together with an adjuvant, right? Now, as I mentioned, to stabilize this peptide, and then of course for the production to be able to do that has been an enormous challenge. And we also worked with another company in the beginning where this was less successful. We started working with PolyPeptide, and we have been super happy with the collaboration. We had a great meeting down at the site in France with the full team, full day, and worked through it. And I think that we came to some very good conclusions on how to progress. And then, of course, we had follow-up meetings very frequently between the teams to work on these steps. And I think that it comes all back to that, right? To get to know the team members and be able to discuss. We also have a partner that we work with in France that are working on the theoretical side of the CMC, you could say, that are supporting us in these discussions, which is very valuable. I mean, also for a small biotech, you can't have everything in-house, right? It's not possible. So you need to work with collaborations. Yeah, we just recently, end of March this year, finished our phase 1b trial where we actually, we saw, first of all, it's important to mention that we had good safety and tolerability in the trial. It's a phase 1, so that's what we primarily look for, right? And then secondary endpoints, we look for immunicity. So we want the body to produce antibodies against this peptide, right? And we succeeded in that. Looks really, really nice. And that's the top things that we looked for. But then in addition to that, we had some explorative endpoints that we also measured, where we looked at cognition, different scales of cognition, and ADCOMS, the most common one, or I shouldn't say common one, the most sensitive one for smaller trials, showed some very promising data in our cohort. And it actually looks like the patients that are on active treatment did not progress in their disease over the 2 years they were on treatment, which, of course, if this holds up when we run it in a bigger cohort, it will be amazing data and can really revolutionize this field. So I'm excited about it and looking very much forward to progress. We also had some very interesting data on biomarkers, particularly one biomarker that none of the other treatments have been able to move, called NFL, or neurofilament light, which is a biomarker particularly for neurodegeneration. And we have some changes there that we think is quite supportive of our other data. Without somebody producing the drug substance, we couldn't do this, right? It's impossible. And there's very few companies in the world that are able to do this type of work. So this collaboration is essential for what we do, of course. And I also like the fact that we are working with a partner that can take us all the way through phase 3 to the market and then onwards, right? Because tech transfer is obviously something that is really challenging for everybody if you have to move. So I think that having a collaboration that can actually support us all the way is great. I think we're very proud of that. We have been able to take it into patients and are ready, phase 2 ready now. I think taking that together, I think it's an enormous achievement, I think, for a small biotech, and most of them fail early, right? So I think that we're in a good position now to take this forward. And then, of course, there's a lot of individual achievements during this process, right? Everybody in the team has really stepped up. We're working on the regulatory pathways right now, which is really exciting to see how they're moving that forward, to have the approvals and hopefully fast track also. So I think that's amazing. I mean, on the CMC side, that we have actually a drug substance now that we can initiate the trial with. and continue. I think that's amazing achievement. Yeah, we are in the process of selecting a CRO now. That's also a lot of hard work that goes into that. So, the clinical development team, I think, there's a lot of people involved and everybody's doing a great job. I think that's the bottom line here. I mean, we have a few things, right? We need to finance this, right? So we're working, of course, on different tracks here. We're working hard on finding a big partner, a big pharma partner that can help us take this through. But we're also working on financing to run the study in our own hands. And I think that these two tracks they can't be mutually exclusive, right? We need to work on both of them simultaneously to see where we end up. You never know what the agenda is for potential partners, and so we have to take the safe road here and try to find a way forward. But what I'm excited about is, of course, to get this started. This vaccine or therapeutic vaccine we're working on is our number one, right? That's what's first in our pipeline. So obviously, to have a collaboration with a partner that is strong, solid, and are able to deliver what we need in time, I think that's essential. Without that, we don't have anything, right? So it's crucial that we have a good working relationship and a good contact, and that we are together driving this forward. And I think also when we met down in France at the site, it was really great to be able to talk to the team also on what we're actually doing, because I got the feeling that a lot of people are working on a lot of projects, right? But they don't necessarily know where this is going to end, So I think for them it was very interesting to see that we're actually trying to change the world here and do something different for people living with this disease.